On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsors, Redshift Bio and Pfizer, I'd like to welcome you to Automated Analysis of Protein Secondary Structure Changes due to Ligand Binding using Microfluidic Modulation Spectroscopy. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now I would like to welcome our presenters for today. First is Valerie Ivancy, Application Scientist at Redshift Bio. Our second presenter is Alison Varghese, Principal Scientist, Structural and Molecular Sciences part of Discovery Sciences at Pfizer. Welcome, Valerie and Alison. And Valerie, we'll turn the webinar over to you.
Hi everyone, thank you for joining us in today's webinar. My name is Valerie Ivancy and I'm an Application Scientist at Redshift Bio. I would like to start today's webinar by introducing Microfluidic Modulation Spectroscopy, also called MMS, and how this technology can be used to determine secondary structure changes due to ligand binding. Powered by MMS, the AQS3 Pro represents the next generation of protein structure determination platforms, providing a novel tool to strengthen the bioanalytical toolkit and provide critical insights during drug development. As a new technology, MMS supports the development of biotherapeutics by reducing the time and cost of development through automation and improving measurement quality of previously undetectable changes in protein structure. Additionally, the data provided by the AQS3 Pro facilitates better understanding of downstream failures and provides quality data for confident, accurate, and timely decision-making. Since the introduction in 2019, the AQS3 has been adopted globally by pharmaceutical companies and academia that continue to push the boundaries to inspire new applications and publications. When looking to characterize protein structure, we have to consider all levels, from primary, secondary, tertiary, to quaternary structure. What you'll notice is we have robust and automated tools for each of these categories that include mass spectrometry, size exclusion chromatography, nuclear magnetic resonance, and dynamic light scattering. However, characterizing secondary structure is more difficult because the available tools include CD and FTIR, which have many drawbacks. CD allows you to look at low concentrations of protein, but is not useful at high concentrations, and has many limitations on the buffers that are compatible, hindering the ability to study biotherapeutics under formulation conditions. FTIR, on the other hand, requires high concentrations of protein and struggles at low concentrations. Additionally, the workflow and data analysis is very manual and time-consuming for FTIR. This is where we come in. MMS is an automated tool that measures secondary structure of proteins over the widest concentration range from about 0.1 to over 200 mg per ml with much greater sensitivity and reproducibility. Analyzing secondary structure is important for characterizing biotherapeutics because structure can give insights into activity, binding, and stability. Specifically, stability is important in choosing formulation buffers that make sure the protein is stable under all manufacturing processes. Secondary structure can also be used to detect aggregation and instability by looking at increases in antiparallel beta sheet formation. Lastly, secondary structure is important and can be used to determine biosimilarity and batch-to-batch reproducibility. The key pillars of innovation in the AQS3 Pro that allow for the high-quality data are highlighted here. First, the quantum cascade laser is much brighter than traditional FTIR light sources, resulting in much higher sensitivity and the ability to detect small changes in structure over a wide concentration range. Next is the fully automated sample handling system that can analyze 24 or 96 well plate. This reduces the hands-on time and human error. Additionally, we incorporated advanced analytics that are intuitive and make analyzing high-quality data much quicker. Lastly, we modulate sample and reference buffer in a flow cell to yield a differential spectrum that enables extremely repeatable measurements and the use of complex formulation buffers. Taking a closer look at the flow cell, we alternate the injection of sample and reference buffer into the laser sampling spot and perform a differential measurement. This allows for the accurate buffer subtraction and high reproducibility. Here's the software data analysis workflow. The system begins by measuring the differential absorbance between buffer and sample, as we saw in the last slide. This spectrum typically consists of about 30 data points and is then interpolated to provide a complete spectrum from around 1600 to 1700 wave numbers. The spectrum is then processed by the software to obtain second derivative. This highlights the features in the spectrum that directly correspond to secondary structure. The spectrum is then inverted and baselined, giving the area of overlap plot. Using this, we can calculate the percent of area overlap to quantify the similarity between samples. This plot is then deconvoluted using Gaussian curve fitting to determine the composition of secondary structure motifs found in a protein sample. Here is an example data set that highlights all the analytical features offered with just one experiment. This is a sample set of BSA incubated offline at temperatures from 25 to 90 degrees Celsius for 20 minutes, then cooled to room temperature for analysis. The raw differential absorbance data is shown in the top left panel and is interpolated at one wave number spacing and converted to absolute absorbance spectrum by normalizing for concentration. As a result of the heat stress, we can see the major peak at 1656 decreases, and there is a clear increase in the signal around 1615. To highlight the system's concentration range, we also ran BSA at 1, 20, and 100 mg per mil concentrations. You can see the quantitation plot in the lower right-hand panel. From the absolute absorbance plot, the second derivative is automatically calculated and can be seen in the top left panel. This lets you see the features making up the absolute absorbance plot. Moving across the top row of spectra, the delta plot is next and is a product of subtracting all second derivative plots from a control you specify. This highlights regions of change across the sample set. The top right panel shows the stability plot that tracks specific wave numbers of interest across all samples. Here, we are showing that as incubation temperature increases, the signal at 1656, representing alpha helical structure, decreases, and 1618 and 1692, representing antiparallel beta sheet, increase. We can also see the onset of change occurs around 65 degrees Celsius, which is in agreement with the melting temperature of BSA. If we take a step back to the second derivative plot for a second, you can see how the similarity plot directly below is derived by inverting and baseline subtracting the second derivative. The percentages of area of overlap are presented in the legend. The 25 degrees Celsius sample was considered the control as that one reports 100%, and as the samples are heated, the similarity decreases. We then use Gaussian curve fitting, as seen in the bottom central panel, to determine the percent secondary structural motifs in the higher order structure plot in the lower right-hand panel. Here, we get the full picture of the structure of BSA at each temperature, and it is clear that increasing the temperature leads to a decrease in alpha helical content and increase in beta sheet content, consistent with aggregation. MMS is a novel, automated tool for looking at protein secondary structure that fits in well with other orthogonal techniques that are currently in use to characterize proteins. For example, SEC for size and aggregation, DSC for melting point and similarity, ITC for binding interactions, HDX for structure such as solvent-accessible regions and ligand binding, and DLS for particle size and stability. Adding MMS to your toolkit provides a more complete overall picture of your biotherapeutic. Overall, the key benefits of the AQS3 Pro are that it can accelerate development by catching instability earlier in the drug development process, and the fully automated workflow saves time and money because we can load and run samples in a 24 or 96 well plate. The improvements in data quality with the AQS Pro allow for analysis of proteins over the widest concentration range available, allowing for analysis without buffer interference and yielding repeatability around 98%, giving confidence in the data quality and ability to observe small changes. Finally, the AQS3 Pro de-risks drug development due to the increases in data quality, allowing confident, accurate, and timely decision-making. With that, I would like to thank you for your time and attention, and I will now pass it back to our moderator.
Thank you very much, Valerie. Let me bring up our next presenter's slides. There, and now we will turn the presentations over to Alison Varghese.
Hello everyone, glad you could join us today. My name is Alison Varghese, and I'm going to take you through some data today that our laboratory has generated using the AQS3 Pro. We will be showing you the results of several experiments using protein generated in our laboratory, and we will be evaluating the effects that ligands and temperature have on these proteins using the AQS3 Pro. So you'll see for yourself what kinds of experiments you can run and the kind of information that you can extract from them. Here's an outline of the experiments that we'll be discussing today. In experiment 1, we'll be running a simple experiment where we quantitate one of our proteins. We wanted to compare the results generated from the AQS3 instrument with the UV280 readings that we generate in-house. In experiment 2, we will be looking at the effects of temperature on an unbound protein. In the third experiment, we will explore subtle changes observed in a protein in the presence and absence of a small molecule. In the fourth experiment, we'll look at the effects of temperature on a ligand-bound protein. And in the last experiment, experiment 5, we'll combine experiments 2 and 4 and look at the effects of temperature on a protein in the presence and absence of a small molecule. So let's get started. In experiment 1, we'll be looking at quantitation of a protein performed on the AQS3 Pro. Accurate protein quantitation is extremely important in pharmaceutical laboratories for many, many reasons. There's assay development, we need to have reproducibility, we do structural and biophysical studies. Most of them are very reliant on accurate protein quantitation. The list just goes on. So here we've removed a protein stock from our freezer whose protein concentration was determined by UV280. And in our lab, we use a Thermo Fisher nanodrop. The protein is about 21,000 Daltons, and we've made serial dilutions from 5 mg per ml down to 0.25 mg per ml using our numbers for this experiment. And we've run replicates for each concentration. We wanted to see how close our readings, our UV280 readings were to the AQS3 Pro. On the right, you can already see from the raw data, the replicates overlay quite well. It looks very good. On this next slide, you'll see the linear regression graph showing extreme linearity with the sample set. Here we've set our 5 mg per ml as the control. As you can see, the fit is very good with the R2 of 0.9999. In the table to the right in blue, you'll see the repeatability of the samples is recorded. And again, where we've set the 5 mg per mil as our 100%. You'll notice the numbers are quite good until you get down to 0.25 mg per mil sample where the repeatability drops, but only down to 95%. In the graph on the left, you'll see another table that notes the input concentration and the calculated fit concentration. This shows us that our UV280 was not so far off the calculations performed by the AQS3 software. I was quite pleased with these results.
In the last slide from this experiment, we show a Gaussian fit graph demonstrating how well the data fits. Also in this graph, we can observe the protein fingerprint of our sample. Here you'll observe on the x-axis the wave number or wavelengths with absorbance on the y-axis. At about 1642, we see a peak which is the signature for beta sheet secondary structure. To the right, at about 1630, we also see a small peak or shoulder. This wavelength is also a signature for beta sheet content. This is more apparent on the right in the higher order structure bar graph, which shows the various secondary structure contributions to the analyzed protein. You'll clearly observe that this protein is primarily beta sheet. Higher order structure is very consistent for samples at half mg per mil or higher. I would also like to mention that while many ligands interfere with various analyses due to their fluorescence, we didn't observe that here. In the lab, we often have issues with protein quantitation in the presence of ligands, high-imidazole, nucleotides, and other additives. Interference really isn't a problem with this technology. I was really impressed with the data quality.
In our next experiment, we'll look at a different protein, which is about 47,000 Daltons. Here we're going to look at the effect of temperature on this protein. We've prepared the protein at 1 mg per mil and 0.5 mg per mil. We've incubated the 1 mg per mil sample for 30 minutes at room temperature, and we've taken the half mg per mil sample and incubated that at 50 degrees centigrade for 30 minutes. And again, our goal here was to observe what happens to the protein upon heating. I just wanted to spend some time on something before I go on. In our laboratory, we use differential scanning fluorimetry, or DSF, as a standard protein characterization tool to assess protein stability and behavior. In particular, we use a nanotempered Tycho NT6, which measures tryptophan fluorescence during a temperature ramp or a protein unfolding. There's many other instruments on the market that measure TI, TM, TAG. Some use dye, some are label free. We have several of these instruments in our lab, but for these experiments that I'm going to describe today, the data will be coming from a nanotempered Tycho. So when you run a DSF, you get a TI, or a temperature of inflection, as a result. This is defined as the temperature at which the protein is 50% folded and 50% unfolded. After a TI determination of an apoprotein or unbound protein, we often will add a ligand to the protein and run it. If the ligand binds, stabilizing the protein, we see a shift to the right to a higher TI. Sometimes it's a significant shift and sometimes it's only a few degrees, but it does give us some confidence when we go to use these ligands that we know they bind, and this is important to know for subsequent experiments. We also use this instrument to evaluate the effects that freeze-thaw, additives such as DMSO or salts, pH, or temperature have on our proteins. It's very useful. In some cases, we've seen things like DMSO cause a destabilization of the protein, causing the TI to shift to the left. DSF is a very powerful protein characterization tool, but it just gives you a snapshot of what's occurring. For this particular protein, you can see that our TI has been calculated to be 46.7 degrees, close to the incubation temperature of one of our experimental samples. On this next slide from experiment 2, the first data that you may notice in the upper left-hand corner is the raw differential absorbance spectra. You can already see a difference between the room temperature sample and the sample that's been heat treated. The heat has significantly affected the protein. The spectra is normalized to generate the absolute absorbance spectra on the right. And you can begin to see the change in the protein fingerprint here. Below this graph is a second derivative plot, which you can observe the changes more clearly. You can see for the room temperature sample in blue, it is primarily alpha helical, which is observed at 1656, while the heat-treated temperature sample is predominantly beta sheet, which can be shown at the right at about 1638. This shift to beta sheet often is indicative of aggregation, which is no surprise. To the left of this data, we show an area of overlap plot derived from the previous second derivative plot. Here you can clearly see the loss of alpha helicity and an increase in beta sheet content in the heat-treated sample, suggestive of aggregation. This plot is used to calculate the percent similarity among the samples using APO room temp sample as a control, and that's shown in this blue table. On this next slide, we have additional data from this experiment. The delta plot in the upper left-hand corner is generated from the second derivative plot seen in the earlier slide. Holding the room temperature sample at 0, you can observe the peak-to-peak changes in the sample. Here we are looking at the 50-degree centigrade sample. In this plot, the major peak at 1656 indicates a loss of alpha helicity, while the negative peak at 1638 denotes an increase in beta sheet content. To the right of this plot is just another way of looking at the delta plot. This is a stability plot, and it shows the changes that occur in the protein when it is heated to 50 degrees. You see the loss of alpha helical content in the orange and an increase in beta sheet content in the green. By default, because of the software, we still get a calculated protein concentration for our samples. At the beginning, we said our room temperature sample was prepared at 1 mg per ml as determined by UV 280. Here, the calculated concentration is a bit higher at 1.1 mg per ml, but that's very close to our UV calculation. More interesting is the heated sample you see here. Our UV 280 determined the concentration for this sample to be 0.5 mg per ml. After heat treatment, the concentration is now only 0.25 mg per ml, according to the AQS3 software. Well, if you remember, our TI for this protein was about 47 degrees, at which half of the protein has unfolded. Lo and behold, it shows that here we've lost just that, 50% of our protein. A nice correlation with our calculated TI. On this last slide from experiment 2, I'd like to show you the last bit of data. That is a nice summary of our experiment where we've looked at the effective temperature on our 47,000 Dalton protein. Higher order structure is calculated by the Gaussian curve fitting on the left. This nicely shows the shift from alpha helical to beta sheet structure in our protein after heat treatment. And this is expected since the temperature in the heat treatment is close to our empirical TI. To the right, our higher order structure bar graph is shown, and you can clearly see that the most significant changes in protein structure due to the increased temperature are again loss of alpha helicity and a gain of beta sheet content, indicating aggregation has occurred. Now on to experiment 3, where we'll look at small changes in a protein in the presence and absence of a small molecule. Here our protein is 25 kilodaltons. Our protein concentration for this experiment is 1.2 mg per ml. And we'll have 2 sample sets in replicate, one APO and one with a stabilizing ligand bound at a 2-to-1 molar ratio ligand to protein. Upon sample preparation, the APO and ligand bound protein will be incubated at room temperature for 30 minutes prior to analysis. The DSF data on the right shows that the APO protein has a TI of 57.3 degrees, which is quite stable already without anything bound. After adding the ligand to the protein, we show that it stabilizes quite well with a temperature shift of 13 degrees, which is quite good. We know it's binding. In this next slide, you can see that the data is well overlaid in the absolute absorbance spectra, which, if you remember, is a normalization of the raw data. To the right, we have a second derivative plot, which begins to clearly show the features of protein secondary structure for this protein. The change between the apo and ligand-bound protein is not as dramatic as in our previous experiment, but you do observe some change. Below, we've again calculated the percent similarity among the samples using the apo as a control. And to the left of this is our similarity plot, clearly showing minor change in beta sheet content at 1638 with a slight increase, albeit subtle. In this next slide from experiment 3, we can show additional data. On the left is a stability plot. And again, from this plot, we can see that there are small differences between bound and unbound protein. To the right, a Gaussian fit plot shows the very subtle changes observed in the alpha helical region between the two sample sets, and a more obvious change in the beta sheet region between apo and ligand bound. The ligand bound is in yellow. These changes are not huge, but they are detectable. For this experiment, here is the finale. I give you the higher order structure bar graph calculated from the Gaussian fit plot. Here we look at the alpha helical content, and we see there's really no overall change. The most change is observed for the beta sheet content, where we see an increase in the ligand-bound samples. This data was not a big surprise. The crystal structure for this protein bound to the ligand has been solved, and it did not show any major structural changes or movement when compared to the apo structure. The AQS3 did a nice job here. In experiment number 4, we will revisit the 47,000 Dalton protein from experiment 2. Here we have the protein at a mg per mil and have added a stabilizing ligand at a 2:1 molar ratio, ligand to protein. We've taken the protein ligand complex and incubated half of it at room temperature and the other half at 50 degrees centigrade for 30 minutes. On the right is the DSF data showing the protein ligand complex to have a TI of about 59.8. In this next slide from experiment 4, you can see from the first set of data in the absorbance spectra in the upper left-hand corner that the data is well overlaid. At the right is the delta plot with the heat-treated protein ligand complex in green, showing change within that sample set. Below that is our second derivative plot, where you can clearly see something's going on in the 1638 region here upon heat treatment, again in green. Our similarity plot to the left clearly shows that with heat treatment, there is some loss of alphalicity and a gain in beta sheet content. Although not overall dramatic changes, but remember, we have a stabilizing ligand bound to the protein here. In our final slide from experiment 4, you can see on the left the Gaussian fit curve. This plot is showing the goodness of fit, and here you can see the obvious change in beta structure upon heat treatment. To the right is our higher order structure bar graph, which is generated from the Gaussian curve. This also shows an increase in beta sheet content, although nothing dramatic, because remember here we do have a ligand bound to this protein. Based on the quantitation data from this experiment, which is not shown, it was evident that after heating the protein to 50 degrees, we lost about half of it, which is no surprise since our 50 degrees is approaching our TI and aggregation has begun. The slight increase in the beta sheet content indicates structural changes upon increased temperature, most likely, again, due to aggregation. In the final experiment presented today, you'll see that it's similar to the previous experiment, except that we will include unbound protein at 2 temperatures, where in the prior experiment we had bound protein at 2 temperatures. So we'll now be including an apo protein as well. Our 47,000 Dalton protein is at 1 mg per ml. We'll prepare apo and ligand-bound protein with the same ligand as used in the prior experiment with the temperature shift of 12 degrees. One of the apo samples will be incubated at room temperature and another at 50 degrees centigrade. The same will be done for the protein ligand complex, one at room temperature and the other at 50 degrees.
The second derivative plot from experiment 5 shows that the apo protein at room temperature and the ligand-bound protein at both temperatures have retained most of their alpha helicity, while the apo protein that's been heated to 50 degrees has almost no alpha helical structure left and is primarily beta sheet, suggesting this sample has surrendered to the heat stress and is most likely aggregated. To the right, the similarity plot shows this a bit more clearly. It is also obvious here that the protein complex at 50 degrees centigrade has also gained some beta sheet content as well and lost a little alpha helicity. But considering the heat stress, it's not that much. The ligand has really protected the protein from denaturation. Below is again the delta plot, highlighting the regions of change in the spectrum with significant change based on whether it goes above or below the dashed line. To the left of the delta plot, we see the same drama with the heat-treated apoprotein.
And finally, our last slide from experiment 5. Here we can see on the left the Gaussian fit curve, which shows this huge secondary structure change in the apo heat-treated sample. From this curve, we generate the higher order structure bar graph, which depicts the changes amongst the 4 different samples very nicely. Again, you can see that heat has significantly stressed the protein without ligand, while its ligand-bound counterpart has been reasonably spared. I hope from these experiments you see how useful this AQS3 instrument can be as a protein characterization tool. You do get a lot of data from one experiment. As I prepared for this webinar, I thought of all the challenges that we face with our projects and how this instrument could really impact them by providing a deeper understanding as to what's going on with our proteins and how different additives, small molecules, buffer excipients, temperature can impact their structure and function. In this slide, I'd like to summarize what we discussed today. We discussed 5 experiments, with the first being a simple experiment looking at quantitation of a protein. In this experiment, we were able to establish the reproducibility among replicates, and protein concentration was determined on samples from 0.25 mg per ml up to 5 mg per ml. Samples at half mg per ml and above showed consistent higher order structure. And as a result, a concentration of 1 mg per ml was chosen for subsequent experiments without further method development. In our second experiment, we looked at structural effects of 50 degrees centigrade heat stress. Protein 2 was analyzed at room temperature and 50 degrees, which is above the known TI, causing the protein to unfold. As a result, the protein lost about half of its alpha helicity and doubled in the amount of its beta sheet content, indicating aggregation was occurring. Our third experiment, we looked at structural effects of ligand binding. Protein 3 was tested with and without ligand bound. Here, there were minimal structural changes observed by binding of the ligand, and this has been confirmed by X-ray crystallography, where we didn't see many changes there either. In our fourth experiment, we looked at heat stress on a protein ligand complex. Here, we complexed the protein and set it at room temperature and 50 degrees. And it showed that the heat stress did not drastically alter its secondary structure in the presence of the ligand. This shows, again, that the ligand provides stabilizing properties for the protein and protects it. In our last experiment, experiment 5, we compared the structural effects on heat on the bound and apoprotein. We combined the results from experiment 2 and 4, clearly observing the stabilizing effect that the ligand has on protein 2. The next steps for future experiments would be methods development for using lower concentrations. We would like to test more complex proteins that require detergents and other additives, and we would also like to evaluate different concentrations of ligand for building titration curves. And with that, I say thank you for your attention.
Thank you very much, Alison. We do have some questions that have come in, and there's still time if you have questions for our presenters. Alison, let's start with you. Raina asks, do you think that the slight change observed in the spectrum of the protein in the presence and absence of ligand could be due to the absorbance of the ligand as opposed to changes the ligand is causing?
Thank you, Elizabeth. Since MMS modulates between sample and reference buffer and takes the differential reading, we included the ligand in the reference buffer as well to account for any absorbance that the ligand might have in this region of the IR spectrum. Hope that answers your question.
All right, thank you. And a question for you, Valerie. How much optimization of the parameters is needed before measuring each sample or spectrum, and also for each sample set?
Thanks. Good question. So the system, as an automated system, has a couple steps before the sample runs that are automated to determine the optimal running conditions for each sample. So the manual sample optimization is very minimal.
Thank you. And another question for you, Valerie. Is there any water vapor subtraction applied to the MMS spectra?
Yes, so in this region of the IR spectra, water is absorbed, but we are aware of where they are, and we have an algorithm that takes care of any water subtraction. In addition, we purge the optics bench to get rid of any water vapor in the gas phase, so that's not a problem for us.
All right, perfect. And Alison, a question for you. Why are the APO-50C replicates much more varied than the APO room temperature replicates?
Well, thanks, Elizabeth. Because the APO 50 degrees centigrade sample lost about half its protein concentration, the signal-to-noise ratio decreased. So this is the type of experiment that might benefit from further methods development to increase the repeatability at very low, maybe sub-half mg per mil samples. Thank you.
Thank you. All right, a question for you, Valerie, from James. What are the upper and lower protein concentration limits for this technology?
Yeah, so the lower concentration limits are around 0.1 mg per mil, and upper, we've gone up to about 225 mg per mil, and that's not to say we couldn't go higher.
All right, and a follow-up question from James. Are any buffers typically found in protein formulations that are incompatible with microfluidic modulation spectroscopy?
Yeah, good question. So we've tested a lot of different buffers, a lot of different excipients, and so far we have not come across a standard buffer that we haven't liked.
All right, and a few people have asked, do you demo the system?
Yes, so due to COVID restrictions, we have been migrating. We are available for virtual demos. In addition, we also offer hybrid demos, where we install a system and kind of do virtual training, but we are very flexible to working with your sites and your restrictions to be compatible with everything.
All right, thank you. And a question for you, Val, as well from Deanne. During the presentations, it was mentioned that this technique allows monitoring a protein in the presence of ligands, such as nucleic acids. Do you have specific examples showing that bound nucleic acids and or oligonucleotides will not interfere with monitoring of a protein?
Yeah, so this is a really hot topic right now, actually, and we've had a couple collaborators that we're working with for different types of applications. As long as we are running the nucleic acid or any type of ligand in the reference channel, we are going to be automatically subtracting that out, and it shouldn't be a problem.
All right, and also for you, Valerie, Wei Lee asks, or comments first, this is a great technique. Can I ask if it can be used for small peptides around 2 kD instead of protein?
Yeah, we've tested a few different small peptides and proteins. For example, we've tested insulin, which is around 5.8 kilodaltons, and we've also tested in a collaboration the amyloid beta peptide, which I believe is small as well. So we are absolutely open to try out your small peptides.
I'd very much like to thank our presenters for today, Alison Varghese and Valerie Avanzi. I'd like to thank Redshift Bio and Pfizer for sponsoring today's event. And most of all, I'd like to thank all of you who came and spent this time with us. We're living in strange times, and we know you're being pulled many different directions. So we're very grateful you chose to come and spend this time with us. So on behalf of Cambridge Health Tech Institute's Global Web Symposia Series, thank you all so very much, and have a great day. Bye-bye.